Thank you, Elisa, for the introductions. So today we're going to be talking about multimodal facility design and really focused around how do you design a flexible facility that allows you to manufacture multimodal therapies all under a single roof. So a quick overview of the ATMP landscape. We've got quite a few different therapies and modalities in clinical trials. These numbers are as of 2019, and they're actually growing and increasing by the week. But you can see predominantly a lot of focus on gene therapy and cell therapy or genetically modified cell therapy, with a little bit of work being done in tissue engineering as well. So I was trying to frame the presentation and why is this important? Well, as the industry is starting to ramp up, I think it's no secret at this point that the ATMP or cell and gene therapy field is growing at a pretty astonishing rates. There are now 17 products that are approved globally, about $10 billion of financing raised in 2019, over 1,000 products in clinical trials, as we saw in the previous slide, and a projected annual compounded growth rate of over 20% through 2023. So it's really an industry that's very much in its infancy state and is at the point where it's ready to, or projected to just take off. Some of the projections for the number of regulatory approvals over the next couple of years are in the hundreds of modalities for cell and gene therapy. So why multimodal? Why is that important at this juncture in the industry? A big portion of it is we are seeing more and more mergers and acquisitions. So ATMP companies have historically been predominantly startup companies, and over the last year, 2019 especially, we've been seeing a lot of acquisition of these small startups by much larger established biotech or pharmaceutical companies. And what that is generating is these multi-product pipelines, where some of these biotech and pharmaceutical companies had typically a limited number of modalities. Now they're acquiring multiple small startups and having to deal with 6, 7, potentially more modalities of product lines. And at the same time, there's some uncertainty in the marketplace as to which modalities are going to dominate. Is there going to be a shift or trend towards one type of modality versus another? So very kind of dynamic environment at this point, where companies are trying to build manufacturing facilities, but they're not quite sure what exactly the future of their pipeline is going to look like. And so an analogy that I like to make, especially when talking about cell therapy manufacturing, is predominantly when you're talking about autologous therapies where it's patient-specific. We are really back to the equivalent of the age of roller bottle manufacturing when you compare it to traditional biotech processes. And if you look at kind of an overview timeline of the monoclonal antibody industry, so kind of traditional biotech, it took really 35, 40 years to get from kind of this being an emerging technology to what we would call modern manufacturing technologies in the mid 2010s, where we're starting to talk about single-use equipment and flexible ballrooms, and really kind of taking these manual processes that were very open and exposed to a lot of risk, and able to finally close them down and automate them and remove your operators out of the clean room to make it a more efficient, more effective, and safer process. So I think there's some lessons that we can learn from the evolution of traditional biotech, which took about 40 years from kind of the infancy stage to modern manufacturing techniques, and compress that timeline for the development of ATMPs and multimodal manufacturing. One of the common issues that we would run into in traditional biotech is there's kind of this antiquated line of thinking about being very risk averse and never wanting to design something that was cutting edge because of a fear of challenges with regulatory approvals. So what you ended up with were projects, and this timeline kind of illustrates this concept, which is as we would start designing a facility for a new client, they would ask us to look around and say, well, what is the latest approved, FDA or regulatory body approved facility? And we want to copy that design. And so, at the time it would take, 4 or 5 years from concept all the way to completion of a facility. And so as you're starting with that starting point, you are effectively designing a facility that is 5 years old. And by the time you've completed construction, you've got a facility that's basically 10 years out of date. So we really want to push the industry to not make those same mistakes and actually compress these timelines and be more forward-thinking as they try to get these new emerging technologies to market in a much faster timeline.
Okay, so today we're going to be talking to you about multimodal facilities. But you'll notice as we go through this presentation that a multimodal facility isn't encompassing of all products. So we want to look at products where it's feasible to incorporate them into the same facility from a regulatory perspective as well as a manufacturing perspective. So we don't want wildly different platforms from a scale or from an equipment technology standpoint. So with that in mind, we're going to be looking at 3 different product types today that we feel are good possibilities for incorporating into a multimodal facility. And these are monoclonal antibodies, gene therapy or viral vector products, and allogeneic cell therapy. So before we take a dive into what the manufacturing of each of these types of products looks like, we're going to give you a little bit of an overview of the science behind each one of those. So first up, we have mAbs, or monoclonal antibodies. So these are engineered antibodies with a single specificity that are produced from a single cell line or a clone. So like Noel mentioned, mAbs have a long history. They originally started back in the 70s as a hybridoma technology, a fusion of mouse and human proteins. But due to immune reactions over the years, they've evolved. So first to a chimeric technology, which reduced the mouse protein content, and then finally to humanized versions that have been in use since the early 2000s. Each monoclonal antibody is engineered to bind to specific antigens, and this allows for treatment of a wide range of diseases, including cancers, autoimmune diseases, and inflammatory diseases. And so, like Noel mentioned before, mAbs have been a mainstay in our industry for more than 30 years, and we don't see them going anywhere anytime soon. But the mAb industry has changed. So we started off with the mAb industry expanding exponentially, and we kept seeing larger and larger stainless steel facilities built to produce these therapies. But times have changed, and with higher titers in use that have improved yields, as well as the widespread adoption of single-use technologies, the big stainless steel facilities are less common. So we're still seeing a demand for mAb production, but where it once may have been a 20,000 liter in scale facility, we're now seeing smaller trains of 2,000 liter bioreactors instead. So our next type of product we're going to be looking at today is gene therapy. And with gene therapy, we're treating a disease that's caused by a genetic defect. So sometimes when a gene is defective, the affected person can't properly code for a protein, and that in turn causes the disease. So with gene therapy, we're providing a copy of that missing or defective gene to a patient so that the patient can express the protein that they're missing on their own. So with gene therapy, we need a way to deliver the corrected genetic material to the patient. You can't just inject patients with DNA. It has a very low uptake rate and reduced functionality. So there are a number of different ways to deliver genes to a patient, but the one that we're seeing most often right now in the industry is through the use of viral vectors. So viruses, by their nature, infect cells and use the host cell's machinery to replicate their genetic material and propagate. So when we use viral vectors, we're taking those same properties of the virus, but we're modifying the virus to include the genetic material of our choice, as well as making the virus's replication incompetent. And there are many different types of viral vectors being used for gene therapy. AAV, lentiviral vectors, baculovirus. These are some of the ones you may have heard of, and each one of these has its own manufacturing platform and production challenges. So when we talk about gene therapy or viral vectors, there are two different types of delivery. So over here on the left, we have what we call in vivo delivery. And for this case, the viral vector that carries the therapeutic gene is going to be injected directly into the patient. On the other hand, we also have ex vivo delivery. And here, the genetic alteration of the cells is going to occur outside of the patient's body before being introduced to the patient. And so ex vivo gene therapy is really a type of genetically modified cell therapy. So one more thing to note about ex vivo therapy is that the patient sometimes may need chemotherapy to reduce the population of cells within their body so that they can better accept the genetically modified cells that are being introduced. And so the final product type we're going to look at today in our multimodal facility is allogeneic cell therapy. So with cell therapies, there are two different types, autologous and allogeneic. Autologous means that a patient's own cells are used. And with allogeneic, we are going to use donor cells. Today, for our presentation, we are specifically going to focus on allogeneic cell therapy. Because this treatment uses donor cells and can be made for multiple patients, larger scales are possible. And the manufacturing process looks more similar to mAb or viral vector manufacturing than autologous cell therapy. So with autologous cell therapy, it's very individualized and a very small-scale manufacturing process, often with manual manipulations performed in a biosafety cabinet or isolator. And that type of processing really doesn't fit well in our multimodal facility, both due to the scale and the equipment that's used. So now that we've talked a little bit about our three different products that we're going to be looking at today, we want to compare and contrast the production for these three different types of therapies. So we've already touched a little bit on MAB facilities. They can be single-use or stainless steel equipment or a hybrid of the two. Cell culture starts in flasks and then moves through a series of bioreactors. They can vary in size. Sometimes production bioreactors are 500 liters, but they could be up to 20,000 liters. This is followed by harvest steps, which can include centrifugation or depth filtration, and then into a series of purification steps, which can include chromatography columns, viral filtration, and inactivation, as well as UF and DF steps. Typically, you don't fill MAB products on site. The product is typically bulk filled, and then it's shipped elsewhere for formulation and fill finish. So with a viral vector facility, the scale is typically smaller. Most of our clients are doing production in 200 to 500 liter bioreactors, but some are planning for a capability of up to 2,000 liters in the future. So due to the high risk of contamination, viral vector manufacturing is almost always single-use equipment. And depending on whether we have adherent or suspension cell lines is going to determine the type of cell culture technology. So with adherent cell lines, you may see growth in T-flasks or hyperstacks, and your production scale bioreactor may be a Paul iCellus. Suspension viral vector production can look very similar to MAB. So think shake flask, wave bioreactors, and then single-use bioreactors. Purification operations are also similar to MAB, including chromatography and UF steps, but we may also see the addition of an ultracentrifugation step. And the ultracentrifugation step is used to separate the viral vectors that have a full payload of capsid from those that do not. At the end of purification, due to the small volumes that are present as well as the limited shelf life, Filling is typically performed within the facility, followed by freezing. And then the final product type we're going to look at today is allogeneic cell therapy manufacturing. And so again, like we mentioned before, since we're using a donor cell line, this is a larger scale process as compared to autologous cell therapy manufacturing. So similar to viral vector, we're going to want to use single-use technology due to cross-contamination risks. And the process typically looks like a series of cell expansion steps using flasks, wave bioreactors, and single-use bioreactors, followed by cell processing steps that can include disposable centrifugation technology like KCEPs, and also TFF. And at the end of our cell processing, we are going to definitely fill on site. So due to the delicate nature of cells, we are going to need to cryogenically freeze them as well before we ship out. And so now I'm going to turn it back over to Noel, and he's going to start us off with design considerations for our multimodal facility.
Thank you, Emily. So what we've done is given an overview of the equipment platforms for the three different main modalities that we were evaluating. And so how does that actually translate into a GMP manufacturing floor plan layout? So the most common one, and I think is pretty well established platform at this point, is the manufacturing of monoclonal antibodies. And you can see in the industry there's probably a wide range of levels of segregation in how the facilities have been designed. Older facilities where process closure analysis was not as available, or closed process technology was not readily available. The segregation was done by facility segregation, which essentially meant isolating individual unit operations in different manufacturing suites and essentially creating a fairly inefficient layout. The more suites, the more HVAC segregation was required, the higher number of airlocks. Over the last 10 years, we've seen operating companies become more comfortable with the process closure and technologies associated with it, which allows you to not to remove the facility barriers and place that equipment into larger ballrooms. So the layout that we've sketched up here is somewhat progressive, I would say. It's not the full ballroom, but it does have one barrier that you'll see on the right-hand side there, broken up into two rooms. And so we have most of our manufacturing equipment is on the left-hand side, with inoculation being done in an isolator, and then all of your cell expansion being done in that suite as well, from the wave bioreactors to your stirred tank single-use bioreactors, and then on to your purification steps. And then we have a physical barrier, which would segregate your viral plus and viral minus steps. So facility layout essentially with two rooms, no airlocks, so fairly progressive. We're saying this is a grade D background with a grade D corridor, and bidirectional GMP flows are acceptable since we don't have any major cross-contamination concerns, especially with the process closure. So our next modality is viral vector manufacturing. And so once again, you can see this kind of two-room layout, something that we're seeing fairly commonly in the industry. What you're doing on the left-hand side is essentially non-viral production. So that would be your cell expansion, cell culture, can be done either in as anchorage-dependent cell lines, or also we're seeing clients trying to move towards the ability to do this with suspension cell cultures. And then as you pass through from the left-hand side to the right-hand side, you're now going to do your viral expansion. So larger stirred tank bioreactors, you inoculate with your viral vector, grow up the viral vector, and then go through your series of purification steps in order to generate your bulk product, which then gets passed on to another location in the facility and gets filled. The primary consideration around viral vector is cross-contamination and the ability for viral replication. So it can still be done in a grade D background if we can prove that we have essentially fully closed processes. But where you'll see a difference is around the flows. We're really looking at having unidirectional flows through the manufacturing suite itself, particularly on the viral side, and also the addition of airlocks for containment and segregation. And the last suite is a layout for an allogeneic cell therapy, and we're really talking about kind of an off-the-shelf cell line, so something that can be grown and manufactured at a large enough scale that it aligns fairly closely with a traditional MAB process or viral vector scale. And once again, this is most of your manufacturing is done on the left-hand side of the suite here. We've got cell expansion or inoculation done in an isolator and then transferred into wave bioreactors and further down into stirred tank single-use bioreactors. Your centrifugation step and clarification steps are done in that same ballroom, assuming once again you have you're able to prove that we have a closed process. And then, as Emily mentioned, filling typically has to be done very, very closely after the purification steps, and part of the reason for that is due to cryopreservation. So most cell therapy products right now, as they're being filled or prior to being filled, are being mixed with DMSO or cryopreservant, and that cryopreservant actually will start affecting cell viability. So there's a very short time frame between the time that you can add your cryopreservant, go through your entire fill operation, and be able to then put that filled bag or vial into a cryo freezer and get it down to cryo temperatures. So once again, kind of this two-room layout, most of the manufacturing being done on the left-hand side, a pass-through into the right-hand suite, which is at that point you've got it's a closed bag, and it is now just going into a control rate freezer until it's brought down to temperature, and then moved over into just a storage cryogenic freezer. You'll notice the difference here is we've got a grade C background. One of the challenges with cell therapies is they cannot be final filtered, and so therefore we like to have a higher grade or higher classification background from a sterility assurance standpoint. So I'll turn it back over to Emily, who will talk a little bit more about our suite.
Okay, so now that we've looked at what an ideal layout could be for MAB, viral vector, and allogeneic cell therapy facilities, we want to overlay these three therapy types into a multimodal manufacturing suite. So the thought here is that we're designing a space that is flexible enough to support all three product types. And so what does the space look like? So to start, each suite is two production rooms. One is a larger ballroom that can support closed processing. And then the second is a smaller flex space that can be varied depending on the product being produced in the suite. So for MAB processing, the smaller flex room could be used for post-viral production. But for viral vector production, the flex room could be used for non-viral cell expansion. Another feature of our multimodal suite is that we're going to design the main processing room or the ballroom to be a grade C space, which is necessary for allogeneic cell therapy manufacturing. If you're running other products, the HVAC can be flexed to run grade C all the time, or it could be designed for grade C and then run at grade D conditions when you're running viral vector or MAB processes. And then for maximum flexibility, we're also going to want unidirectional flow in our layout. So we're going to have a separate supply and return corridor and segregated airlocks for personnel and materials entering the suite and for waste and personnel as they exit the suite. So now we're going to go through and take a look at our multimodal facility with each different product type in it and look at how different processes can function in the same space. So our first process is MAB production. And you can see here that we've gone with an open ballroom concept from inoculum prep through viral filtration. And we can do this because the process is going to be functionally closed. All of our transfers are going to be closed. Product transfers and additions are going to be made either via tubing wells or aseptic single-use connectors. You'll also notice a cytocentric isolator in the upper right-hand corner, which is going to allow us to not have a separate suite for inoculum prep. So the open processing manipulations that are typically completed in biosafety cabinets within an inoc prep suite can now be located within our ballroom space. So typically, the industry has still wanted to see a separate environment for post-viral purification steps, and so we've segregated the final UF step as well as bulk filling into our flex space. So next, we're going to take a look at what viral vector manufacturing can look like in our multimodal suite. So here, we're going to use our small flex space for cell culture prior to transfection, and this way we can keep non-viral and viral cell cultures segregated. Moving into our ballroom space, we have our single-use production bioreactors where transfection is going to occur, followed by purification steps, including depth filtration, chromatography, TFS, and viral filtration. You can see that our transfection cocktail is also going to be prepped in the ballroom, but within a small isolator. And then there are additional process steps that you sometimes see in viral vector production that we're not showing here. So, for example, ultracentrifugation. But we could modify this facility to accommodate that as well. You just may also need additional small isolators for aliquoting the product into the centrifuge tubes, and then for removing the product as well after ultracentrifugation is complete. And then our final layout is for allogeneic cell therapy production. And in this case, we're going to use our larger ballroom for all of the cell production and processing. So similar to viral vector and MAB production, we're going to have an isolator that we use here for inoculum prep for those small volume manipulations that are typically open. This is going to be followed by production in a series of wave bioreactors and then single-use bioreactors. For harvest, we're going to use a KSEP, which is a type of single-use centrifuge, followed by TFF. And then, like we had mentioned before, it's really critical to do filling for allogeneic and autologous cell therapy right away. So we do have a filling isolator within our ballroom as well so that we can go ahead and fill our product. In our flex space, we're going to use that for vial inspection as well as freezers, both controlled rate and cryogenic. And one consideration that we haven't touched on earlier is that these freezers are going to need liquid nitrogen supply to them. So having some adjacent mechanical space here for liquid nitrogen dewars or having a liquid nitrogen supply to these spaces is going to be advantageous.
Thank you, Emily. So now I'd like to discuss a little bit of our case study, which is focused around how to build or design and build this multimodal flexible manufacturing facility. And so we have many client requests now that are asking about multimodal manufacturing. There's maybe quite a handful of facilities out there that do some level of multimodal manufacturing, but the way it's currently done is with dedicated suites. So if somebody has a MAB process and a viral process, there's some questions about whether or not those can coexist in the same facility, and the answer typically is yes, with the appropriate level of segregation, proper design, proper operating procedures, there are multimodal facilities out there. However, what we're starting to see become a more common request is this idea of building facilities with suites that are now not dedicated and have the ability to be campaigned from one mode to another. The premise of building a multimodal facility with dedicated suites does not really give you the level of flexibility that matches the industry's needs at this point with these fluctuating pipelines. So we've taken a first pass at laying out a concept for these modular units and generating a manufacturing floor plan that is capable of campaigning between these different modes. And what that allows you to do is get better utilization of your space, especially not really understanding, which of the products in the pipeline are going to move faster through clinical stages and what your future manufacturing requirements are going to be. So as you design this facility, and Emily touched on it a little bit, you superimpose these 3 suites into what we were kind of referring to as the mother of all suite, this very flexible suite. You have to take kind of the higher constraints of all 3 different modalities and superimpose them into a suite that allows you to run that flexible process. When you put them together, you can see kind of a general floor plan here with 8 units. And in this configuration, you could potentially be running a variation of different product combination from week to week or month to month as you campaign from one suite to another. This type of manufacturing configuration can be particularly advantageous to contract manufacturing operations who have very little control over what product lines they will be manufacturing and what modalities are coming down their pipeline. So as you take this suite, obviously all the functionalities are not necessarily required for all the different modes, but you're going to operate essentially in the same fashion regardless of which mode you have that is currently being manufactured in that suite module. So even though unidirectional flow, for example, is not required for all the modalities, once you start building this out as an overall manufacturing core, you would very likely just operate the suites, every suite, as a unidirectional manufacturing suite. And then something Emily touched on was HVAC classification, and there is some flexibility in designing a system that could potentially handle a grade C background and be ramped up to a grade D background, but very likely, in all practicality, it probably makes sense to just operate as a grade C, design as a grade C, operate as a grade C, and then either just continuously manufacture under grade C operations, even though you don't necessarily need it for some of your modalities because you have process closure, or you could design it to a grade C but validate to a grade D if you don't foresee using that suite for allogeneic cell therapy manufacturing for a period of time, and then revalidate it to a different level if you require that higher background. And so when we look at HVAC, not knowing exactly which products are going to be in which one of these suites, once again, you design it with the proper level of airlocking for segregation and containment. You segregate some of these manufacturing suites, which may not be required for each modality, but once you get into viral vector manufacturing, for example, you probably do not, or it's not advised that you would want to recirculate the air from the viral vector expansion suite into your cell expansion suite and potentially contaminate it. And you definitely would not want to recirculate air in between your different suites with modalities that are potentially not compatible. So it is, in a sense, a little bit of a worst case design from a facilities and mechanical equipment standpoint, but it does provide a much higher level of flexibility when it comes to the actual manufacturing operations. And some of the other considerations you want to make sure that are taken into account is creating enough space so that you can roll in and roll out the proper equipment for those different modalities. In some cases, you may be able to reuse some of the equipment across different modalities and campaigns, assuming that it's single use and you've done the proper line clearances, but you'll also need to be able to account for pulling equipment out of the suite, rolling other equipment out of the suite. So some careful consideration around the size of material airlocking and how do we get equipment in and out of the facility. And then Emily will tell us about some additional facility considerations, maybe not focused around the manufacturing core itself, but more around the actual entire facility design.
Thanks, Noel. Yes, so Noel just presented on what a multimodal facility with multiple suites can look like, but there are definitely some other facility considerations we should take into account that are specific to these facilities. So as we look at campaigning different products in the same suite, we're going to want to have the ability to perform some type of room fumigation, or VHP. So this doesn't necessarily have to be a built-in system, as those can be quite expensive, but we at least want our rooms designed to be VHP capable and have the option to contract out VHP of the suites in between campaigns. We also haven't mentioned solution prep up until this point, but each of these product types has a fairly high consumption of buffers and medias. And so we'll need a space to prep as well as stage all of these buffers and medias before we move them into the individual processing suites. For QC, we're used to seeing ATMP facilities having larger than typical QC support areas. And for multimodal facilities, there can be some overlap in staff and technology, but each of these different product types is going to have some unique testing requirements. And so as a result, our QC areas are going to need to be flexible as well to support a varying mix of product types, as well as the QC functionality to support them. Another option could be to outsource a portion of the QC testing, particularly the more unique tests, and then just have in-house testing for those tests that are shared among different product types. So staffing and facility headcount is also going to be kind of interesting for these types of facilities. It's definitely going to grow, and the staffing in this case is going to be more similar to a CMO. So operators are going to have to be cross-trained to run multiple different product types. And then finally, warehousing, material movement, and control of incoming and exiting materials are all going to be critical for multimodal facilities, especially because what you're manufacturing today could be very different from what you're manufacturing a year down the road. And Noel touched on this a little bit earlier, but since we have so many different technologies, you're going to be moving single-use equipment in and out of your rooms. So we're also going to need some pretty sizable equipment storage space so that as you campaign, you have a place to store the equipment that you're not using. So why are multimodal facilities so attractive? Well, it's because many of the facilities that we're designing and building right now are for therapies that aren't yet approved. Many of them are in clinical trials. And if you have a product in phase 2 clinical trials and it fails and you're building a facility for it, having a flexible facility that you can use to manufacture other products that are coming through your pipeline is very attractive. It also gives you the ability to support legacy products and also to adapt to changing market demands. And one of the trends that we've noticed is that often multiple companies are racing to develop a therapy for the same condition. So having a flexible facility like a multimodal facility is going to let you adapt to changing market demands.
And one of the things that we want to mention is this is by no means the silver bullet or the answer to every industry challenge out there. There are some modes that do not lend themselves very well to being campaigned, whether it's from an equipment standpoint or a segregation standpoint. One of the modalities we have not talked about much in this presentation, but there's starting to be a lot more interest being generated in the industry is around plasmid manufacturing, for example. And when you're talking about microbial fermentation, we are now talking stainless steel tanks and CIP infrastructure and things that are more fixed in place and do not really allow you that flexibility to roll equipment in and out of a suite. So just wanted to make sure that we touched on that, and that by no means is this flexible suite just allow you to completely eliminate the need for any sort of dedicated manufacturing, but that it does allow you or create a lot more flexibility around some of the more common manufacturing modes out there.
Thanks, Noel. So as we wrap up the presentation, so when we started out the presentation, Noel gave us an overview of MAB processing and highlighting how far we've come over the last 30 or 40 years. And so likewise, as we close out the presentation, we wanted to look at the ATMP industry and where it's come over the last 30 or 40 years as well. So looking back, it's been 40 years since the first stem cell lines were derived from mouse embryos. The first FDA-approved gene therapy trial happened 30 years ago. And then in the last decade, the industry has really sped up. The discoveries being made have really accelerated. So CRISPR was discovered in 2013. 2017 was a really big year. It saw the approval in the U.S. of both the first genetically modified cell therapy, a CAR-T called Kymriah, as well as the first approved gene therapy, Sparks' Luxturna, for vision loss. And as we look at all the progress that research and scientists have made in this field, we want to make sure that the facilities that we're building for this industry are going to take us where we need to go over the upcoming years. So we need to build facilities that are flexible and adaptable for different technologies and the products that are going to come to market. And so this is why we need multimodal facilities. Thank you.